Hello everyone, I am Mahesh and in this video I will be discussing about modern concept of rational drug design, challenges of a medicinal chemist and the process of drug discovery. In the olden days, drug discoveries were made by random sampling of plants and plant samples. These plant sampling is being done based on ancestral evidence. Many such drugs are being discovered. For instance, opium, ephedrine, and so on. The accidental discovery of penicillin led to the discovery of various drugs by screening large number of antibiotics from bacterial and fungal sources. Presently, thousands of new organic chemicals are prepared and are examined for their biological activity. Drug design is an interdisciplinary science which involves the study of effects of biologically active compounds on the basis of molecular interactions in terms of molecular structures or its physicochemical properties involved. Drug design is not only based on modern computational chemical techniques, but also uses the sophisticated knowledge of disease mechanisms and receptor properties. Any drug development process must be proceeded through several stages in order to produce a product that is safe, efficacious, and has passed all regulatory requirements. The discovery often begins with a target identification, choosing a biochemical mechanism involved in the disease condition. Drug candidates discovered in academic and pharmaceutical or biotechnological research laboratories are tested for their interaction with the drug target. Up to 5,000 to 10,000 molecules for each potential drug candidate are subjected to a rigorous screening process which can include functional genomics and or proteomics as well as other screening methods. Once scientists confirm the interaction with the drug target, they typically validate that target by checking for the activity versus the disease condition for which the drug is being developed. After careful review, one or more lead compounds are chosen. The drug development includes various steps such as product characterization, formulation, delivery, packaging development, pharmacokinetics and drug disposition, preclinical toxicology testing and IND. This preclinical toxicological testing and IND application includes acute studies, repeated dose studies, genetic toxicity studies, reproductive toxicity studies, carcinogenic studies, toxicological studies, and so on. After all these preclinical studies, bioanalytical testing is being done, and later, the various stages of clinical trials are being conducted, like phase 1 clinical studies, phase 2 clinical studies, and phase 3 clinical studies. After the phase 3 clinical studies, the drug or the molecules will be considered as NDA. These NDA drugs are being registered or sent for registration in the various regulatory bodies like FDA. Once the FDA review is being done, only then the drug or the molecule is sent out to the market. And this whole process of drug discovery takes between 10 to 17 years for each drugs. It requires the synthesis of more than 10,000 molecules to get 10 molecules into the clinical trials and one new drug. And the cost of the drug discovery and the preclinical studies will be more than $355 million. And the total cost would be near $2,000 million. When it comes to the pharmaceutical R&D, there are hundreds of different disciplines working together. So, coming to the research and development in the pharmaceutical industries or in the pharmaceutical sectors, it is a multidisciplinary team and it's not being done by a single person or a single team. So the drug development process starts from the identification of the disease, isolation of the protein, finding a drug effective against the disease protein, and then preclinical testing, scale up and formulating, file IND, go for human clinical trials, file NDA, and then FDA approval. So whole of this, as I have told you, it takes 10 to 15 or 17 years. And the drug design comes over here. In the finding the drug effective against the disease protein is where we can find the drug designing mechanisms or the drug designing process. The drug designing process includes genomics, proteomics, and biopharmaceuticals, the potentially producing many more targets and personalized targets includes in genomic, proteomics, and biopharmaceuticals. And then comes the high throughput screening. Over here, we screen up to 1 lakh or 1 million compounds a day for activity against a target protein. Then we have virtual screening using a computer to predict activity. We have combinatorial chemistry where rapidly producing vast number of compounds. We do molecular modeling, that is using computer graphics and models with to help the improve of activity. And then we have in vitro and in silico ADMA models where tissue and computer models begin to replace the animal testing. So this whole process comes under the drug design. In this slide, we'll be discussing about the various disciplines involved in the drug discovery. So these are the concepts what is to be kept in mind while we are studying drug discovery. Coming to the structural activity, pharmacodynamics and pharmacokinetic properties, we have to have a thorough knowledge of biochemistry, pharmacology, immunology, enzymology, and so on. The finding out of novel molecules include combinatorial chemistry, synthetic chemistry, and the pharmacokinetic properties also should be understood with the knowledge of physical chemistry. So when we have to study about the safety and in vivo activity, we have to have the knowledge about metabolism, the safety assessment, pharmacology again, and pathology. The drug designing mechanism involves molecular modeling information technology, which includes mainly the use of computers or AI, which is artificial intelligence. After all these things, the intellectual property comes into the picture where we study about the patent law. The challenges of a medicinal chemist in the drug discovery includes the cost of the discovery process, the time taken for the discovery of medicines or the drug molecules, and fierce competition. The history of drug development begins like this. In the year 1909, the first rational drug designing was being seen. The goal of this first rational drug design was to get safer syphilis treatment than atoxyl. So, in that times, Paul Ehrlich and Sakario Hata wanted to maximize toxicity to pathogen and minimize toxicity to human, that is the therapeutics index. They found out Selversan, which was replaced by penicillin in 1940s. First successful attempt to relate the chemical structure to biological action quantitatively through quantitative structure-activity relationship was being discovered by Hans and Fujita. Drug design is an integrating developing discipline portending an era of tailored drug involving the study of effects of biologically active compounds on basis of molecular interactions in terms of structure or physicochemical properties. A lead molecule is a prototype having desired biological action with or without undesirable characteristics. Undesirable characteristics include high toxicity, other biological activities, solubility, metabolic problems. Drug discovery without a lead are very few. In mid to late 20th century, the medicinal chemists had a very restricted knowledge about understanding the disease state, biological structures, process, drug transport, distribution, and metabolism. Medicinal chemists used this knowledge to modify chemical structure to influence a drug's activity, stability, etc. So over here in this slide we can see procaine, which is a local anesthetic, and a slight modification in the structure of this procaine leads to procainamide, which is an anti-rhythmic drug which has a completely different class of activity. Modern drug discovery includes or involves the identification of hit, lead discovery, lead optimization, and identifying the drug candidate. In medicinal chemistry, an optimization of those hits to increase the affinity, selectivity to reduce the potential side effects, efficacy, potency, metabolic stability to increase the half-life, and oral bioavailability. Once a compound that fulfills all of these requirements has been identified, the process of drug development can continue, and if successful, clinical trials are developed. The various approaches of drug discovery include serendipity, chemical modification, screening, and rational drug discovery. In the serendipity, taking an example of Alexander Fleming, Fleming studied staph, but contamination of plates with airborne mold noticed bacteria were lysed in the area of mold. A mold product inhibited the growth of bacteria that led to the antibiotic penicillin discovery. This we call it as a serendipity or accidental discovery, which we cannot depend on every time. Hence, our efforts as medicinal chemists includes the following, like chemical modification. Chemical modification includes homolog approach, that is, homologs of lead are being prepared. The method of homology modeling is based on the observation that protein tertiary structure is better conserved than amino acid sequence. Thus, even proteins that have diverged appreciability in sequence but still share detectable similarity will also share common structural properties, particularly the overall fold. Under molecular disconnection, the term disconnection relates to breaking a carbon-carbon bond of a molecule to generate shorter or simpler fragments. A good disconnection must achieve the greater simplification of the target molecule. For a complex molecule, this basic disconnection process is repeated until the target is reduced to simple starting materials. The complete set of disconnections and functional group interconversions for a specified target molecule is what constitutes a retrosynthetic pathway or plan. Under molecular addition, an addition reaction occurs when two or more reactants combine to form a single product. This product will contain all the atoms that were present in the reactants. Addition reactions occur with unsaturated compounds generally. The fourth way of chemical modification includes isosteric replacements, which includes the interconversion of isomeric structures leading to difference in potency or the activity. The next approach is screening. In screening, testing a random and a large number of different molecules for biological activity reveals the lead molecules. Innovations have led to automation of this synthesis, in which we use combinatorial synthesis and testing through high throughput screening. Example for that one is Prontosil is derived from a dye that exhibits antibacterial properties. In rational drug discovery, which starts with a validated biological target and ends up with a drug that optimally interacts with the target and triggers the desired biological action. Under rational drug designing, there are two kinds: pharmacophore based approach and structure based approach. In pharmacophore based approach, a pharmacophore model explains how structurally diverse ligands can bind to a common receptor site. Pharmacophore models can be used to identify the de novo design or virtual screening novel ligands that will bind to the same receptor. This pharmacophore based approach includes quantitative structure activity relationships, 3D quantitative structural activity relationships, and qualitative structure activity relationships. The structure based approach or the structure based drug design attempts to use the structure of the protein as a basis of designing new ligands by applying the principles of molecular recognition. Selective high affinity binding to the target is generally desirable since it leads to more efficacious drug with fewer side effects. Computer-aided drug design uses computational chemistry to discover, enhance, or study drugs and related biologically active molecules. The most fundamental goal is to predict whether a given molecule will bind to the target and, if so, how strongly. So in this slide, you can see like where and all the computer aided drug design is involved in the process of drug discovery. Molecular mechanics or molecular dynamics are most often used to predict the conformation of small molecule and to model conformational changes in the biological target that may occur when the small molecule binds to it. Semi-empirical ab initio quantum chemistry methods or density functional theory are often used to provide optimized parameters for the molecular mechanics calculations and also provide an estimate of the electronic properties of the drug candidate that will be influencing the binding affinity of a drug. So the computer aided drug designing over here, the main advantages of this is the cost of the drug discovery is reduced to a great extent. At the same time plays a very important role. So this computer aided drug designing reduces the time to really around 6 to 8 years. Accuracy is more in the computer aided drug designing. Reproducibility is high, that we can observe the database screening. The screening is reduced, very less manpower is required, and ultimately we have the complete information about the disease. So these are the advantages of computer aided drug design. Thank you.